The microtubule cytoskeleton is essential for correct intracellular organization, cell division, and differentiation. During mitosis and meiosis, microtubule assembly is locally upregulated by the chromatin-dependent Ran-GTP pathway. Local production of Ran-GTP around chromosomes liberates proteins that contain a nuclear localization signal from the inhibitory action of importin, a nuclear transport receptor. Among them is TPX2, an elongated protein having prominent roles in microtubule formation around chromatin. Both over- and underexpression of TPX2 perturb microtubule organization, leading to genomic instability, and mutations in TPX2 are correlated with high metastasis frequency in cancer patients. In addition to several microtubule binding regions, TPX2 interacts with numerous binding partners. The TPX2 nuclear localization signal interacts with importin, and this interaction suppresses TPX2 binding to microtubules and microtubule nucleation. However, the structural basis of the effects of TPX2 on microtubule stabilization and nucleation, as well as their regulation by importins, is not yet understood. In this work, we determined the cryo-electron microscopy structure of a central region of TPX2 bound to the microtubule surface. We observed a novel microtubule binding mode with two flexibly linked elements of TPX2 binding the outer microtubule surface across both longitudinal and lateral tubulin dimer interfaces. The microtubule binding region of TPX2 directly overlaps with the nuclear localization signal and the importin interaction motif. Our results reveal that this interaction mode is critical for microtubule binding and facilitates microtubule nucleation. The binding mode of TPX2 explains its selection for GTP-bound tubulin at the tip of microtubules and suggests that TPX2 may slow down hydrolysis, further contributing to microtubules stabilization. This novel microtubule binding mode provides a structural explanation for how TPX2 suppresses microtubule dynamics and stimulates microtubule nucleation, and how the Ran-GTP gradient can regulate TPX2 microtubule interaction through importins.